Good morning, good afternoon, and good evening to all of you who have taken the time to join MedGenome's webinar, Elevate Your Research with Advanced Analysis of Bulk and Single-Cell Transcriptomics Data. Thank you so much for joining us. My name is Aditya Pai, and I will be your host for today's session. Our agenda for the day will include introductions, an overview of genomics and its importance to bioinformatics, single-cell transcriptomics and its applications, as well as how important bioinformatics is and can elevate your research in this area. We will also have a Q&A session. Please use the Q&A feature to type in your questions, and we'll do our best to respond to these after the presentation, and if not, respond to you by email. Our two speakers for today include Dr. Kushal Suryamohan and myself. Dr. Suryamohan is a Director of Bioinformatics Services at MedGenome and leads the Commercial Bioinformatics Services team. He oversees the optimization as well as development of pipelines for NGS data analysis. Previously, he worked at Genentech, where he focused on analyzing long-read optical map sequencing and chromatin capture technology data to generate high-quality de novo genome assemblies. He's also involved intricately in applying high-throughput NGS-based functional genomic screens of oncogenes to characterize oncogene function in cancer. Dr. Suryamohan is widely published in high-impact journals and holds a PhD in biochemistry and a master's degree in computer science from the University of Buffalo, State University of New York. Myself, I'm VP for Corporate and Business Development at MedGenome. Most recently, I was VP for Sales at Genuity Science, where I led sales and business development for sequencing, oncology assays, and disease cohorts for the biopharma industry. Prior to that, I was a global sales leader at IBM Watson Health for Watson Oncology and Life Sciences products, where I was also appointed as a member of the IBM Industry Academy as a healthcare and life sciences industry expert. I've published numerous articles in scientific journals and industry reports, and hold an MBA from the Schulich School of Business at York University in Toronto, which is where I live, and master's degrees in medical genetics from the University of Newcastle upon Tyne, UK, molecular genetics from McGill University, Canada, and genetic counseling from Brandeis University. Let me first begin by saying what an interesting time we live in. This is 20 years after the publishing of the human genome, and this is a very special time. As many of you have surely seen with the graph on the left, the cost per human genome has continued to fall dramatically. From what was an unsustainable rate at the turn of the century, we can now sequence a human genome for less than $1,000. And this is far better than even Moore's law, which is used in computing. It's an extraordinary time indeed. And as data was referred to as the new oil as it related to even electronic health record data, now sequencing data can be considered the new oil. When you look at the Human Genome Project and the fact that it took 240 machines in 2 years to go through an entire human genome, which was 3 gigabytes, it now takes a fraction of that time, and you can compute over 48 human genomes with sequencing of 6 terabytes of sequencing data in a week. Truly, there's a plethora of data, but that also means that insights are required from that data. Genomics has various applications, and these applications are from basic research to translational research. If you look at genomic data from DNA, you can sequence the entire human genome or the exome, or look at targeted genomics and single-cell genomics. You can look at the epigenome and look at methylation effects or protein DNA or protein RNA complexes, as well as chemical modifications. You can look at transcriptomic data, which is a big focus of our talk today, with genome-wide expression analyses, single-cell transcriptomics, as well as in situ transcriptomics. And then you can also look at the gut or the microbiome analysis and analyze how the microbiome might also be impacted. This is all possible through a whole range of different tissue types. It can be whole blood, PBMCs, it can be cell-free DNA, target tissues, or even purified cells, tissue cells. And the result of this is a massive explosion in this entire industry, which can range from target discovery, where you're identifying new interactions or associations that are genetic in basis and their mechanism, constructing polygenic risk scores with genome-wide association studies, looking at prognostic data with biomarker discovery to offer prognosis for a particular condition, looking at pharmacogenomic markers as responses to treatment, stratifying different patients based on their molecular profiles, repurposing drugs using unique features of those drugs that are completely different than even the reasons that they were originally designed and intended for. And then ultimately diagnostic implications, which is the discovery of diagnostic molecular profiles. In short, genomics is an integral part of biomedical research. So much so that at the recent NIH conference, Eric Green put forth bold predictions for human genomics for 2030. We've highlighted in blue the ones that have a significant impact on data. Let's just take an example of that. Number two, the biological features, functions of every human gene will be known. For non-coding elements in the human genome, such as knowledge, such knowledge will be the rule rather than the exception. Underlying this is the basic implication that data is going to be at a premium, and this data will require bioinformatics for the analysis as well as interpretation. So how do you handle such large amounts of genetic data? This is akin to Amazon delivering a 1 billion, 3 billion piece puzzle at your doorstep, and you being asked to, in fact, reconstruct the puzzle, which is the human genome. Bioinformaticians are key to enabling this. And in fact, they help extract value from such vast amounts of sequencing data with a very skilled bioinformatics team. This is an interdisciplinary team that manages large amounts of data, managing the database where such data is housed, look at algorithms and pipelines that can help with computing complex genomic data coming from a variety of different types, as I just showed you. Very important, collaborating and communicating with research stakeholders, principal investigators, interpreting that data, visualizing that data, and problem solving, working hand in hand with the principal investigator, developing diagnostic tools, and ultimately realizing value, which is extremely important to most biological experiments. Having massive amounts of data is not as useful unless you can actually draw insights from that data. MedGenome has a world-class bioinformatics capability. We have a track record of working with diverse omics data sets of various types. We can support your needs at any stage of the research project lifecycle, right from experimental design to publication. We have creative domain experts, and I stress the word creative because you do need to be creative in terms of the different types of experiments and analysis that can bring new insights and life to your omics data. We can act as an extension of your team, but this requires trust, the fact that you've done it before reliably, credibility with your track record, as well as reproducibility and provenance, as well as efficiency and rapid turnaround times. We prevent bottlenecks with our ability to scale very rapidly with fast turnaround times and work in parallel with your research teams, allowing you to draw insights at the same time you're doing your own analysis. MedGenome is a leading omics diagnostics research and diagnostics partner. We are located in Foster City in California, as well as Bangalore, India, and support 4 key businesses. Our research services business focuses on working with top research organizations and pharma companies to generate omics data, whether it's in the US or the rest of the world or in India. Our bioinformatics service, as you will see from Kushal, offers advanced analysis and visualization of omics data. We are also the #1 provider of genetics diagnostics in India, serving over 150,000 patients a year and offer more than 475 different genetic tests. And in the growing part of our business, genomic medicine, where we have the largest and deepest South Asian clinical genomic data set with research collaborations with key clinicians, research groups, and access to patients across key therapeutic areas. This is an area that is vastly underrepresented in most population genomics databases that have a distinct slant towards more Caucasian populations, where an Indian genomic data set would add tremendous value as a result of high consanguinity as well as endogamy. Let me move into an area that is of our focus today, which is single-cell transcriptomics and its applications, and spend a little bit of time on that. This is one of the most remarkable slides and probably one of the early insights into how the field of single-cell began. Brenner, Sulston, and Robert, who all got the Nobel Prize, described the cell lineage for part of the nervous system, and in 1983 published a cell fate map, which detailed the lineage of every cell in C. elegans. During development, 1,090 cells are generated, but precisely 131 of these are eliminated by apoptosis or programmed cell death. This results in an adult nematode worm comprising of 959 somatic cells. Can you imagine what we can do with 30 trillion human cells and the thousands of cell types? How do you study them? These insights were originally found in the C. elegans, where we have learned some fundamental aspects of development biology, how cells differentiate, how cells die, as well as many other stochastic features of these cells. There's been exponential scaling of single-cell sequencing technologies in the past decade. The ability to go inside a single cell out of those 30 trillion, find and lyse that cell and obtain cDNA from that, and find a method to actually create a library and amplify it and sequence it. That is something that is remarkable. And you can see that in the last 5 years, there's been a tremendous amount of publications as well as technologies commercial that have come to market that have indeed revolutionized the way we look at medicine. You can look at single cell RNA sequencing, and this is by far one of the most explosive areas of growth. And that's because you can identify different cell types, lineages, cellular heterogeneity, and look at single cell level sequence screens, as depicted by being on the front page of the most eminent publications in research. Single cell sequencing provides a very unique process, and compared to bulk sequencing, the best analogy I can give you is bulk sequencing is more like a milkshake, versus single cell sequencing is the individual fruits that go into that milkshake. Whereas you have a heterogeneous population of cells, some experiments are designed to look at bulk sequencing of that transcriptomic data. However, you can only look at average gene expression, nothing wrong with that, except that you cannot understand cellular heterogeneity at the level you might want to. With single cell sequencing analysis, you can look at per cell gene expression, you can look at clusters of those cells, and can understand better cellular heterogeneity. A very good example of this would be with tumor oncology, with tumor biology as well, where you look at a tissue such as a tumor. A tumor has a number of different cells and what we refer to as the tumor microenvironment. You can isolate and sequence individual cells as shown by the 4 different colors. And for each of those, you can compare gene expression profiles of the single cells. You can then branch these and break them up into a principal component analysis, and that will give you some unique insights into how those individual cells interact not only amongst themselves, but also between each other. The application of single cell technology is crucial, and one of the key areas is in the immune system that we see. So when we look at comparing immune microenvironment across tissues, or even the regional immunity in tumors, there's tremendous amount of data that you can generate through single cell analysis. You can look at the study of the immune system. You can identify normal immune cell types, clonotypes, as you'll see shortly as well. And this offers a tremendous amount of insight into areas such as virology, areas such as immunology, as well as oncology. And this has come to the forefront with the COVID-19 pandemic, where we have often tried to wonder, why do individuals respond differently to COVID viruses versus some other individuals? With tumor oncology in particular, bulk genomic analysis and expression profiles of clinical specimens have shaped much of our understanding of cancer in patients. Human tumors, for example, are very intricate ecosystems with diverse cells. These include malignant cells, immune cells, stromal types of cells, whose precise characterization is often masked by bulk genomic methods, which are really still a dominant method used. With single-cell genomic technologies, these have appeared as a powerful way to dissect the human tumor level at the resolution of individual cells, and then provide a very compelling approach to deciphering cancer biology. Our single-cell solutions that we offer at MedGenome include various sample types. The sources are from fresh cells, frozen cells or nuclei, fixed cells or cryopreserved tissues. With tissues, we perform tissue dissociation with single nuclei as well. That is another new area that we have started focusing on. Our solutions include single-cell gene expression, single-cell TCR, BCR, which is really the focus of our talk today, single-cell attack-seq, site-seq, as well as cell hashing workflows. And we use the 10x Chromium technology with the Tcr plate-seq technology for library solutions and preps, as well as our Illumina solutions through the MySeq and NovaSeq for sequencing. And as you will see from Kushal, the discovery that comes out of the TCR, BCR, bulk, as well as single-cell analysis will offer some very interesting views of how the data can be analyzed and looked at. But let me just also take a pause and take a moment to look at the immune repertoire and the profiling of the immune repertoire. This is an extremely difficult and complex process, but it's also a very high sensitivity method detection which is required to uncover the repertoire and the diversity there. The diversity of the TCR repertoire is analyzed by looking at the unique number of CDR3 that you see in the middle of your screen. And these CDR3s are important because when specific T cells with unique TCRs expand by binding to an antigen, there is a selective expansion of CDR3. When one such T cell clone dominates, what we call a clonotype, the clonotype gains importance into understanding immune response. We have about 10 to the power 10 unique TCRs, and the diversity of this VDJ recombination is why we have such a strong army of T cells and B cells behind us that help us protect ourselves against infection. This T cell diversity is what we call the T cell repertoire. And the implications of this are tremendous. They can be therapeutic in terms of looking at identifying specific antigens that are specific to TCRs or creating TCR engineered cells. From a diagnostic perspective, you can assay TCR clonality. And from a monitoring perspective, you can look at treatment monitoring by assays looking at the TCR repertoire as well. Let me also show you this figure where massive strides have been undertaken in the past few years in how we sequence and analyze TCRs. Single chain sequencing technologies have been instrumental in broadening this understanding of this adaptive immune system and will continue to be extremely important. However, it doesn't describe the combinatorial heterodimeric pairing of receptor subunits. As you saw in the previous diagrams, there are subunits with the alpha and beta chains as well as heavy and light chains. But this type of pairing is not something that can be done and is necessary for complete characterization of repertoire diversity, as well as for downstream experiments that look to produce complete antigen receptors or characterize the function. There's been a lot of interest in exploiting the advantages of high throughput sequencing with paired antigen receptor sequences and for single cell sequencing technology, where you can look at a single lymphocyte resolution for large cell numbers. Once you encapsulate these in emulsion droplets, reverse transcription can proceed separately for each bead, and each is coded with an individual unique barcode. And subsequently, you can use these to assemble a heavy chain, light chain fusion amplicon, which is then sequenced as a single unit. With our antibody discovery platform, as Kushal will show you, this is a method that we can use for the purposes of antibody discovery. Let me just pause there and turn it over now to Kushal. I'm going to stop sharing my screen, and Kushal, over to you.
Thank you, Adi. Just let me share my screen. Okay, so thanks, Adi, for that very nice introduction to single-cell transcriptomics and the myriad of applications that have really exploded in the last few years. So what I'm going to do is walk you all through some of the bioinformatics that's involved in single-cell genomics data analysis and more specifically, immune repertoire analysis. So many of you are familiar with 10x Genomics and the out-of-the-box analysis software that they provide for analysis of single-cell genomics data and single-cell immune repertoire sequencing data. Typically, this means that they have an end-to-end solution where you start with your DNA or cell suspension. You go through your barcoding and library construction, and then you sequence the libraries, and then you have an analysis suite from 10x that includes the long range and cell ranger pipelines, which are the workhorses underneath the visualization software. And you use the Loop browser to visualize your results. And that's shown here in the screenshot where you have some functionalities to analyze this data. But really speaking, this browser is very limited in its capabilities and functionalities, even being able to tweak some of the parameters that you end up with this clustering is not really possible. And some of the more advanced analysis for you to be able to slice and dice your data is not possible with this out-of-the-box software. MedGenome has been working tirelessly in the last few years using its extensive bioinformatics and biology expertise to build solutions that add value to clients and research projects by building an analysis platform which we call Mango, MedGenome Analytics for Genomics, which is a customizable framework for genomics analysis. What this means is that we can take platform agnostic software solutions that have been hosted in containers such as Docker or Nextflow or Conda packages, or even inbuilt in-house developed software pipelines. They all go into this framework, and that can be deployed either in the cluster or on the cloud, which hosts a database at the back end. And we integrate all of these pipelines, and we generate reports using MATLAB, R, Python, and we provide high quality reports. And the good part about this framework is that it is scalable and a reproducible platform for data analysis, visualization, and we provide all of our reports for any research project using this Mango platform. And so you would have reports that are in HTML format that you can browse through pretty conveniently in one place and go through the data, the QC reports, the visualizations, and the tables. I'm just showing you screenshots from an RNA-seq pipeline where you can click on each of these sections here and go through the reports. And so it's really a one-stop solution for any bioinformatics pipeline that you may be interested in. We've taken this and built a platform that can now provide some of the advanced analyses that I was mentioning is not currently offered by 10x's software. In addition to 10x's genomics output files in the Loop browser, we can now provide advanced additional analyses such as differential gene expression, clustering of cells using Tcr or Umap, uh, Umap algorithms, and looking at the clusters of cells resulting from the selection of the most informative features from your single cell multi-dimensional gene expression data, and being able to annotate the cells that have been identified in these different clusters using a machine learning based automation, uh, or using manual curation to identify these clusters based on known gene expression markers. We also provide analysis for pseudotime uh, and lineage tracing, which can help you identify the differentiation of different cells into their lineages. We also do advanced visualizations of individual gene markers and look at the breakdown of the expression levels across the different cell types that we've identified, either manually or through automated curation. In terms of immune repertoire analysis, we provide a number of different metrics and visualizations that are currently not really part of any sort of standard software package that's out there. We provide summary statistics on the clonotypes, the current clonal homeostasis. We provide an ability to track clonotypes in longitudinal time series experiments. We look at different clonal diversity estimate matrices, and we also look into the CDR3 and VJ gene usage statistics shown here. And more recently, we've started providing support for integrating multi-omic data sets, which is really sort of the most uh, useful way of looking at your data, where you can interrogate multiple aspects of your cell. You can look at gene expression data, you can combine it with immune profiling data, you can combine it with epigenomic data at a single cell resolution. And so what do these outputs from the single cell analysis I just mentioned look like? We initially look at QC, which is sort of an integral component of single cell analysis, where we look at different aspects of the QC, such as the number of features, the number of barcode counts in your samples, and the contamination measured in terms of mitochondrial reads that map to mitochondrial DNA. And so our bioinformatics team carefully looks at this QC data, and following that, we decide on a number of parameters for data normalization and feature selection. Essentially, you want to identify the most variable set of features that can then be used downstream for scaling and dimensionality reduction using principal component analysis. So this sort of initial QC is critical for any downstream step. Once we have the principal components identified, then we do a number of visualizations downstream, either TSNE or UMAP. UMAP has now superseded TSNE for visualization of single cell data. And so the UMAP clustering here shows you the segregation of the cells into different clusters. And using either machine learning, reference-based annotation, or manual curation based on known markers of interest, we can then identify and annotate these different clusters into cell types, be it T cell subtypes or B cell subtypes, or any other cell types. It could be pancreatic cell subtypes, and so on. We also provide visualizations that break down the proportion of these cells into their subtypes, which is another informative way of looking at the clustering and the proportion of cells present in your sample of interest. And this could be very important when you're looking at a disease versus normal state. We then also perform differential gene expression between the different clusters. This is, slightly different from bulk RNA-seq, where you can look at differential gene expression between samples. Here, you can actually look at differential gene expression between individual clusters that you've identified within your population of cells. And we can take the top markers from each of these clusters, and this could really aid in biomarker discovery. You can identify a particular gene signature that's specific to your cell type of interest, can be used to identify rare cell types and their markers. And so there's a number of different applications that go along with just the first two slides that I showed you here. And so in addition to all of that, we provide a number of other different outputs from our single cell analysis. And all of this, as I mentioned, is delivered through our Mango platform. So it begins with FASTQ files that are shared via FTP, an HTML web summary report of all of the data that includes 10x outputs, as you see here, where I'm hovering my mouse over. We also provide advanced analyses such as customizable flexible t-SNE plots, Umap plots. We do automated cell type assignment or even manual cell type assignment. And then we provide marker-based gene heat maps and differential gene expression analysis. We also do pathway analysis of the differentially expressed genes, and a detailed pathway enrichment analysis report is also provided along with this visualization. For bulk RNA-seq, we also provide splicing analysis that we're also looking at providing for single-cell data as well. Moving on to the immune profiling part of single-cell genomics and transcriptomics in general, there are two flavors of immune profiling. One is TCR profiling, and the other is BCR profiling. And TCR analysis has sort of, really been prominent in the last few years with the development of two different approaches. One is the bulk approach, and one of the most frequently used applications is provided by Takara Bio, where you can amplify TCR alpha and beta chains, and you can sequence the repertoire that is present in an individual or in a tissue of interest. We've also developed proprietary assays for profiling the TCRs, not just in humans, but also non-human primates, such as monkey and the macaque. And this is very important, as these assays can now support drug discovery and development efforts, but also vaccine efficacy trial, because TCR clonotypes can expand in a disease, and the ability to clone the receptors and validate them functionally, expedites your drug discovery or vaccine efficacy trial as well. And so we've developed these assays to essentially take samples of interest and amplify the VDJ region of each TCR chain. Incidentally, we can also amplify the delta gamma delta gamma TCRs as well. And so with the libraries prepared, we sequence them on a MySeq, and then we deliver all of our reports using Mango, even for the TCR analysis. In parallel, we've also developed a high throughput monoclonal antibody platform, which we call HitMap, which uses 10x single cell BCR profiling. And one of the advantages of this platform is that it now enables rapid discovery of thousands of antibodies using the single cell BCR platform. And it really bypasses the old hybridoma-based antibody discovery technology, making this technology more rapid and cost-effective, because hybridoma-based technologies are slow, low throughput, labor-intensive, and can be quite expensive. We've now sort of developed this platform to produce to provide services that include BCR profiling of species outside of 10x's standard human and mouse samples. So we can now provide BCR profiling for horse and rat as well. And it's sort of important to mention here that we've actually built bioinformatics capabilities to be able to profile the horse and rat antibodies, because these are again not standard out of the box solutions. So we've actually built bioinformatics capabilities to be able to identify BCRs in the horse and rat. And this actually has a very relevant application. We've recently been able to use this platform to identify over 5,000 SARS-CoV-2 N protein antibodies, and we've actually tested and validated many of these antibodies, and we've now created a solution for identifying COVID-19 antigens with high sensitivity. And so just to summarize, for your TCR or BCR profiling requirements, MedGenome can provide an end-to-end workflow, beginning from sample all the way through advanced bioinformatics analyses. And so in terms of the analysis outputs that we provide for TCR or BCR sequencing is a whole which slew of data and reports, including information on the clonotypes, full length, partial, productive, and for single cell data, we provide information on the number of successfully paired antibodies, the frequency of the productive clones, and also summary statistics on the top clones. Uh, we look at sharing of clonotypes between samples, qualitative sharing of clonotypes, and quantitative analyses as well of the clonotypes. We also look at the proportion of TCRs within each sample. We look at fold change expansion or contraction. We look at V and J gene usage to see if there is any preference for a particular V or J gene. We look at the diversity of the clonotypes using a number of different indices to look at the overall richness of your clonotype space in your sample of interest. And so I'm just going to walk you through a few of those summary statistics and visualizations that I just mentioned. What you're seeing here are essentially what we like to call summary statistics on any TCR or BCR analysis. The first plot here shows you the overall proportion of clonotypes within each sample. You would expect the larger the fraction in a bar plot you see, it just means that a greater number of clonotypes occupy the space in the overall repertoire. You also look at rare clonotypes, which could be interesting for particular diseases or any other biological question of interest. We also look at the overlap of the clonotypes between your samples. And again, this shows a qualitative way of looking at how clonotypes are shared between disease pathologies uh, or compared to a normal tissue. We also look at in-depth CDR3 frequencies, V and J gene frequencies, shown here as a bar plot that shows the preference for each V gene. And similarly, we look for D gene usage as well between samples. In this case, we're looking at 2 samples, but we can look at any number of samples and their V gene usage or J gene usage. And we look at the correlation between the V and J gene usage here. We also look at distribution of CDR3s. CDR3 length can have a role as well. And so we look at the distribution of the CDR3 lengths and the preference for a particular V or J gene in terms of their frequencies for these different lengths. And overall, we provide a summary of all of the clonotypes that were identified through our bioinformatics analyses. And these summary tables are very nice when you want to look at the top frequent clonotypes and what proportion of the clonotype space does a particular clonotype use, what is the V gene it uses, what is the D gene, what is the J gene segment, and what's the constant region, and sort of the business end of the TCR or the BCR, which is the CDR3. In addition, we provide full-length clonotype sequences and all of the visualizations that I just showed you. This is another representation of looking at V and J gene. This is more akin to the RNA-seq analysis, where we look at V and J gene usage patterns. The Shannon diversity is one of the diversity index metrics that you can use to look at diversity of your clonotype. We also look at any preferential motif enrichment in the CDR3s, and this could again be a qualitative analysis that could help aid your research. Another way of looking at V and J gene usage is through these circus style plots, where you can look, if there are 2 samples that are given individual stimulants, is there anything that you can say qualitatively and quantitatively about VJ gene usage? And so you can see here, stimulant A, there's a particular J gene that has a preference for a few of the V genes, but upon stimulant B, there's a significant expansion of 2 or 3 clonotypes across the V and J gene segments. And so the circus plot enables this as well. And so this is another useful way of looking at your data. We also provide correlation maps, which allows you to see qualitatively if there is any increase in sharing of a particular clonotype across 2 samples. So in blue are all the clonotypes unique to the sample on the y-axis. In yellow are clonotypes that are unique to the x-axis, but in red are the interesting clonotypes that are actually shared between 2 samples. And so again, these are visualizations that you can really use to understand your data better. We can also track clonotypes longitudinally. So in this case, you're seeing 3 patients and clonotypes that have been identified in each patient. And if you're integrating it with single cell data where you have different clusters identified, you can look at how the clonotypes are represented in each cluster across the samples. And you can even look at treatment groups, or you can just track clonotypes by time in samples. So you can basically track the change in frequency of these clonotypes across samples or across treatment conditions. And you can also look at the overall proportion of the clonotypes and how they're changing in time or by samples, basically, if there's a greater expansion of a particular clonotype in one sample versus another sample. And this essentially identifies interesting clonotypes that could be further validated or studied downstream. And finally, the power in single cell technology is to be able to assay multiple data points. And here I'm showing you integration of single cell RNA-seq data with TCR or BCR data. And what I'm showing you here is one patient who's been treated with a particular drug. And we're looking at RNA-seq data and single cell RNA-seq data. And after performing the clustering using Tcr, as I mentioned earlier, we arrive at these different clusters, but we now also have information on the TCR clonotypes, and we want to see if there's a particular expansion of one or more TCR clonotypes in response to a drug treatment. And you do see a significant expansion of TCRs um, and the clonotypes post-treatment, and be able to identify these can potentially give you candidate cancer vaccines. And so we're now at this stage where single cell multi-omic data generation and analysis is really possible. And at MedGenome, we provide the ability to integrate multiple sources of single cell genomics data, be it protein information, such as cell surface protein information, such as methods using site-seq, or single cell RNA-seq, or immune profiling, single nuclear RNA-seq, which is really from single nuclear sequencing, single cell ATAC-seq, which is information on chromatin accessibility, or epigenetic modifiers, histone proteins. And we are now able to integrate and study at the same time different dimensions of genomic data. Besides single cell genomics, which was the sort of the focus of this talk, MedGenome also provides support and domain expertise in a number of other genomics areas, including bulk transcriptome sequencing, metatranscriptomics. We also have expertise in whole genome sequencing and exome sequencing using targeted capture panels. We do HLA typing, metagenomics as well. And as I mentioned, epigenomics using ATAC-seq, ChIP-seq, cut and tag, methyl-seq. We obviously provide immune profiling support. We also have extensive experience in de novo genome assembly and annotation for non-model organisms and model organisms. We also have built capabilities for imputation in our South Asian specific population cohort, as Adi mentioned earlier. And more importantly, we can build custom analyses pipelines, visualization tools, and integrate multi-omic data sets, in addition to a number of other capabilities that our bioinformatics team has. And so with that, just to summarize the sort of the webinar today, we gave you hopefully a high-level overview of some of the real-world applications of single-cell genomics, specifically immune profiling. And MedGenome can help you with this type of application and can analyze your data beginning from sample collection and sequencing to bioinformatics analysis. Given our deep domain knowledge, we can really be considered as an extension of your research team. And we have a number of different models that we're happy to answer, in terms of engagement, be it on a per project basis or as a full-time employee, depending on your project needs. All right, so thank you for your time. And at this point, Adi and I are happy to take any questions, be it about single cell genomics or anything bioinformatics related, really.
Yes, so thank you very much, Kushal. And there are a few questions that have come up. And please, if there are additional questions, please use the Q&A tab and at the bottom of your screen next to participants, and you can please type in any question. The first question is, can you please talk to the pervasiveness of single cell technology in vaccine discovery, virology, and oncology?
Yeah, I think there's been a lot of discussion now. So that's a great question. Traditional vaccine development, traditional vaccine development, usually builds upon, you know, a laborious process of identifying the immune repertoire, the the ability to be able to even identify converging sequences of TCRs or BCRs previously was not possible with bulk sequencing, but now you can really do that with single cell genomics, where you can actually identify paired antibody sequences, and to be able to clone these sequences and actually test them really gives you more confidence in what you've discovered, and if that can be used to uh, potentially give rise to cancer vaccine candidates. We also recently worked on gallbladder cancer recently, where we were able to identify potential neoantigens and use them as cancer vaccine candidates. And so the ability to use TCR or PCR sequencing, in this case TCR sequencing, for cancer vaccine and vaccine development is also pretty critical.
Yeah, thanks, Kushal. And I would just add that I think with the advent of bulk TCR, BCR, as well as single cell approaches, you're able to get to a level of CDR3s, as you saw with some of the outputs that Kushal showed you, in a way that was just never possible before. So when you're looking for an immune response, you're looking for immunogenicity, I think this technology is certainly going to help and become much more pervasive as we go along, simply because I think the ability to sequence that level of data, which was a rate limiting step years back, has reduced, as has the advent of newer technologies with whether it's 10x Chromium versus Takara as well. A second question was, what is the difference between next generation sequencing and second generation sequencing? So I can answer that by saying that next generation sequencing is highly automated. It has the ability to produce sequencing data at a very large scale and can be used from virtually any sample type. And there's a commercial system through Illumina or Thermo Fisher, or if you're looking for long range reads through PacBio or Oxford Nanopore, that can give you those long range reads as well. Right? And that's the fundamental difference in the scalability of such a solution, a next generation NGS solution versus before. I don't know, Kushal, if you want to add anything to that.
Yeah. So essentially, next generation sequencing is also traditionally sometimes referred to as second generation sequencing. So all your Illumina based sequencing technologies were referred to as second generation. First generation was really Sanger sequencing. And now we're into the third generation of sequencing, which is really long read sequencing, such as PacBio, Oxford Nanopore. So hopefully that answers your question. Yeah. It's sort of synonymous that NGS is basically high throughput sequencing, as Adi mentioned.
Another question was, do you offer your analysis service as a project-based basis? So I can answer that, Kushal, initially, and I can say that yes, absolutely, we offer our resources on a project basis, or if you're looking for full-time resource to work on your projects as an extension of your team as well. And Kushal, if you want to add anything to that.
No, I think you've pretty much covered it. Yes.
Okay. Then are
And
you
I
able
think
to
another, sorry, just to add to that though, is, we realize that bioinformatics analysis is highly iterative in nature. And so we are highly flexible in working with, our clients and research partners in doing iterative research, because you eventually need to slice and dice the data in different ways to be able to really arrive at, significant conclusions that can make more sense out of the data.
And are you able to do tissue dissociation and single nuclei sequencing? The answer is yes, and very recently we have optimized the method in our lab and can use tissues for dissociation, and therefore isolate single cells and do the analysis from the sequencing of those, as well as from single nuclei. Can you talk about multi-omic capabilities? So Kushal, do you want to answer that?
Sure. So we have recently begun providing support for multi-omic data analysis. As I showed you in the last few slides, one of the more frequently requested sort of multi-omic data analysis is integrating single cell RNA-seq data and single cell TCR or BCR data. And what that allows you to do is look at expression profiles of specific TCR or T cell immune markers, but also look at the TCR clonotypes and how they can correlate with the expression data. we can also integrate single cell attack-seq data and single cell RNA-seq data, which is also now possible through 10x's multi-ohm analysis and multi-omics kit, basically.
Great. Do you see this technology being used in CAR T cells or cell-based therapy approaches? And the answer is yes. If you have engineered CAR T cells that you are using, certainly a really important part of those CAR T cells, once they're injected back into a patient in a clinical trial, would be to look at what is the microenvironment in which they are being injected, what is happening at the single cell level. So single cell can be extremely useful in addition to bulk sequencing approaches as well. And sometimes this is where it can get, I would say, interesting, is if you're also looking at the pure genomic data as well through blood and looking at whole genome sequence. So a combination of whole genome sequence along with selective use of single cell approaches, whether that be looking at the immune repertoire, T cell or B cell repertoire, or looking at 5' 3' gene expression, those can all be used in tandem. So it's very relevant to that type of a technology where you're actually injecting an engineered CAR T cell and looking for what kind of a response you're getting. What technique do you use for tissue sorting? I might have to get back to you on this one specifically. Yeah, we'll get back to you on this one in terms of the specifics of tissue sorting. But we have used.
I mean, I can take that. I mean,
Yeah.
tissue sorting might not be the right
Yeah.
question. I think it's more
Tissue
dissociation, and then we don't do any sorting. We typically expect the client the samples. If we do expect cell sorting, then it has to be done before we get a hold of it, because we do not do fax sorting at our end. Typically, sorting is done using markers of cell surface markers of interest.
How do you integrate multi-omics data? Do you represent each omic data as separate plots or multi-omics as a single plot?
So it can be a bit of both. So as I showed you in my slide, you can look at both data on a single plot. For instance, the TSNE plot, which is single cell RNA-seq based analysis, overlaid with the TCR plot as well. So it really depends on the requirement and the application.
Okay.
Or what technique do you use for droplet? So for droplet sorting, we use the 10x Chromium, but we also have plate-based methods for sorting cells into single cells.
I think that's all the questions I had. Any other questions? We're happy to stay on for a few more minutes if there are, and if certainly, please continue to ask the questions on your Q&A.
We're always happy to take your questions via email as well.
Absolutely. Please feel free to email us, and we'd be happy to send you those responses back as well. And of course, we hope that the session has given you some interesting insights into how single-cell transcriptomic data, especially as it pertains to immune profiling and looking at both bulk single-cell TCR, BCRs, is highly relevant in today's environment. And no better place to look than the COVID pandemic that has hit us, as well as looking at oncology data, where this has proven to be quite beneficial in getting a really deeper understanding than was ever possible before. So we appreciate your time. We'll stay on for a few more minutes if there are any more questions to be typed. And again, thank those who want to drop for their time. We appreciate you taking the time through your day, whether it's morning, afternoon, evening. It's great to see such a great group across the world, really. Can we inculcate this into early cancer diagnosis? Let me just see if I understand this question. So if you're asking if your single cell 10x method, where we're creating the gems, beads, and attaching a barcode and sequencing after amplification sequencing, those give insights into a single cell. Obviously, if you have cancer tissue or tumor, that is comprised of a variety of different types of cells, and therefore you can get some really interesting data from that. When you're referring to early cancer detection, that is typically done on, if it's done non-invasively, it's done through cell-free DNA, circulating tumor DNA that you see in blood. And there are methods that look at minimal residual disease in those cases after treatment, or early based detection methods, such as those that look at methylation patterns in particular, right, that are offered by companies such as Grail. So it is possible, I would say, depending on what you're looking at, early cancer diagnosis through blood-based approaches has gained a lot of popularity. And that's exactly for the reason that you're able to detect cancers using methylation changes in the circulating tumor that is seen in blood, and you're able to identify that. Right. I'm hoping that answers your question. Kushal, I don't know if you wanted to add anything more to that.
No, I think you've hit all the points there. So, yeah.
My previous question was two separate questions. How do you integrate multi-omics data? Do you represent each omic as a separate plot or multi-omics? The second question is answered. Would like to know about the first one. What do you mean by integration and how is it done?
So integration is essentially you're combining the two data sets. It doesn't mean you're combining them sort of, so to speak, like physically. You're looking at patterns of correlation between the two data sets. So that is what we mean by integrated analysis, where you're able to take one data set and parse out information from, let's say, RNA-seq data. You're looking at, so I can give you the example of single cell data, right? So because you're able to simultaneously assay the same cells for gene expression and also T cell receptor data, you can actually identify each cell by its barcode. And so you have TCR data for for that cell, right? But you also have gene expression data for that same cell. And so in that case, you're actually looking at multiple data sets, but in a single cell. So you can ask questions like, are known T cell markers highly expressed? And what does that mean in terms of the TCR clonotype information? Do I see a higher frequency of clonotypes for cells that express this particular gene marker? And so that's the sort of multi-omic integration that we do. Hopefully that answers your question. But there's the other sort of integration which doesn't have to happen in the same space. For instance, you can have transcriptomic data in a particular tissue, but you also have open chromatin accessibility data. And you could ask questions like, are genes that are near open chromatin regions highly expressed? Are they not as highly expressed as one would expect? And are there transcription factors bound near these open chromatin regions that could be driving higher expression? And if so, what are the transcription factors? And we do that typically through motif enrichment analysis. And so that's another example of multi-omic data analysis, which doesn't have to happen simultaneously, but can still be used to integrate and look at multi-omic data sets.
I think that concludes all the questions that were being asked. Again, we'd like to, on behalf of MedGenome, I'd like to thank all of you for attending the session and appreciate the thoughtful questions as well as your participation. We look forward to doing more of these and certainly bringing advanced novel technologies as well as analysis capabilities to the forefront as well. And again, thank you very much. Have a great afternoon, evening, morning, wherever you might be. Thanks.
Thanks, everyone.